All right, so we're just about wrapped up. I'm going to go, this next section I'll go through really quickly. We're just reviewing some of the structures in the cell. This should all be reviewed to you guys, but I want to go over it really quickly. So the nucleus, obviously, we have in most eukaryotic cells, genetic information is protected by a double membrane known as the nuclear envelope. Prokaryotic cells do not sequester their genetic information inside a nuclear membrane. So like bacteria, for example, they have plasmids. There are different ways that a cell can store its genetic information, but in eukaryotes, we like to sequester it safely inside the nuclear envelope so that we can carry our functions safely inside there, away from all of the degradation that occurs in the cytoplasm, for example. So the nucleus contains machinery for replication and transcription of DNA and the splicing of RNA. So the translation of mRNA occurs in the cytoplasm. So remember, if we're transcribing DNA, we're making that mRNA, it leaves the nucleus after being spliced, of course, and then translation occurs in the cytoplasm at the ribosomes. And it's not pictured here, but it's worth mentioning of the structure known as the nucleolus, which is located inside the nucleus. Nucleolus actually means little nucleus, and that's what it kind of looks like. It just looks like a little dot. It's not present in this diagram, but you just have to know that it produces ribosomes. It's basically a ribosome factory, and it's located inside the nucleus as well. So we have some ribosomes. They are not bound by membranes, so technically they're not considered organelles, but they are super important structures for all cells, basically. So it's the site of protein translation, and it's in the cytoplasm. And I say cytoplasm because I'm contrasting from the nucleus. There are also ribosomes that are on the endoplasmic reticulum, so they may be free in the cytoplasm or attached to the RER. So RER ribosomes produce proteins that usually end up with the following fates. They usually secreted from the cell, they become a cell membrane protein, they become a Golgi, lysosome, or an ER protein. So when they're made in the ER, they're usually on that path, they'll go to the Golgi. They're usually being packaged for something to be sent to another structure or to be secreted from the cell. Free cytoplasmic ribosomes make proteins that are usually headed toward peroxisomes, toward the mitochondria, the nucleus, or will stay in the cytoplasm. So the rough endoplasmic reticulum is a system of folded membranes that gets its rough appearance from the presence of ribosomes. So it's in the cytoplasm, but these ribosomes are sort of studded on the walls of the endoplasmic reticulum, and that's why we call it rough, because it looks rough. And the rough endoplasmic reticulum, it synthesizes proteins from membranes and export from the cell. It's also continuous with the nucleus. So a lot of things that come out of the nucleus will pass through the rough endoplasmic reticulum without ever actually being in the free cytoplasm. So proteins will often move from the rough endoplasmic reticulum directly to the Golgi, where it is packaged into membranous vesicles. So it's sort of like a conveyor belt from the nucleus to the ER, and then it goes from the ER to the Golgi. The smooth endoplasmic reticulum, you can see it looks a little bit different. It's still the same thing. It's just a system of membranous sacs, basically. It's folded membranes, excuse me, but it does lack the ribosome concentration of the rough ER. So because it doesn't have ribosomes, it's not actively involved in protein processing, but it does play a role in steroid synthesis, for example, in the gonads, and it can also aid in the degradation of toxins in the cell. So Golgi that are in liver cells will help purify the toxins in that cell. And you can see they're right next to each other, same membranes, just there are no ribosomes on the smooth ER. Then we have the Golgi apparatus, also known as the Golgi bodies, or most simply just the Golgi. And those are a group of membranous sacs located outside of the endoplasmic reticulum. Sometimes people liken them to a stack of pancakes, anything like that. It's just sort of like a smashed stack of folded membranes. So the Golgi may modify proteins from the ER. They may sort proteins to send to specific final destinations, and they can also synthesize certain macromolecules like polysaccharides. Proteins coming out of the ER, the endoplasmic reticulum, remember, will enter the Golgi from the cis phase, travel unidirectionally through the membranes, and exit out of the trans phase toward the cell membrane. So when things move through the Golgi apparatus, it is usually unidirectional. There are certain examples of where proteins move the opposite direction in a retrograde fashion. But for the most part, everything that is working normally is moving unidirectionally through the Golgi apparatus from the cis face to the trans. And you can remember trans means across. So if you're ever confused between which is cis and which is trans, just remember that the cis is the one that is facing the entry of the protein, which is toward the endoplasmic reticulum. The trans face is across from where the protein is entering. So it has to cross the Golgi to get to the trans face, which means across. So remember, the trans face is facing outward. Then we have the mitochondria. So mitochondria are small organelles composed of an inner and outer membrane, and are the site of oxidative phosphorylation. So while the inner membrane is impermeable, to the diffusion of polar substances, the outer membrane has large pores and is not very selective. The inner membrane, the site of the electron transport chain, is folded into structures called cristae, which increase the surface area. So you can see the cristae right here. And you could see they're folded. That's the inner membrane, and then the outer membrane is a lot more smooth. It's not folded. So mitochondria possess their own genome. The endosymbiotic theory is not something you need to know too much about, but it's just the theory that states that mitochondria were once most likely individual prokaryotic organisms that were taken up by a larger organism. And there's a lot of evidence for the theory. One being that mitochondria exhibit their own genome. They have this double membrane. The inner membrane sort of looks very familiar to gram-negative bacteria. So it's very likely that if these were budding out of another organism, that second membrane was formed around it due to a situation like that. But you don't have to know too much about endosymbiotic theory. Just know that that's an explanation for why the mitochondria is sort of a little bit different from the rest of the organelles, specifically because it does have its own genome. And because it has its own genes, the mitochondria, they exhibit complete maternal inheritance. So when the sperm fertilizes the egg, all of the mitochondria that eventual organism will have come from that egg, come from the cytoplasm in the egg, because the cytoplasm in the eventual body comes from the mother cell, and so do all of the mitochondria. So the next structure we're going to talk about are the centrioles. Centrioles are a barrel-shaped structure that play an important part in cell division. So if you look to the right, you'll see they do sort of look like little barrels. So when the cell enters mitosis, the centrioles are responsible for creating the microtubules that make up the mitotic spindle. We just spoke a little bit about that, microtubules, before. Paracentrioles will move up to the poles of a dividing cell and pull apart the sister chromatid pairs to begin anaphase. So as you can see right over there, these mitotic spindles are moving to the poles and they're going to pull them apart. And that's basically the main function of centrioles. That's definitely what you need to know them for. Okay, and then we have one final review and we are finished for the day. So in which of the following places would you most likely not find a ribosome? Okay, so in which of the following places would you most likely not find a ribosome? So the correct answer is B, the smooth ER. The nucleolus, as we mentioned before, is a ribosome factory. That's where they're produced. The rough ER is the final location of many ribosomes. They're studded on the walls of the rough ER and they produce proteins there. And D, we also have a bunch of ribosomes, floating around freely in the cytoplasm as well. So you would find them there. The only place you wouldn't find them is the smooth ER. And we call it the smooth ER specifically because of the absence of ribosomes. It gives that membrane a smoother look. So B would be the correct answer for that one.